Everyone.
Welcome back.
Yeah, we are here for another workshop in this series. I'm Felipe Matias. I'm talking here from Brazil. Yoo-hoo! And with me here,
I'm Claire Hagen. I am from Agriculture Canada.
and we are working this project called PhenomeForce. And in this series, let me share my screen. Here we go. And we always start with a short presentation explaining about this project. This workshop, we are talking about principles, protocols, and pipelines for drone phenotyping. It is a partnership with Drone to Phenome Group. And who we are, Claire?
We're PhenomeForce. So if you found this video and you want to see more, we have a whole YouTube channel, and we are a community looking to connect people working in phenomics and empower early career researchers. And we're trying to build a library of online resources in phenomics, and we're offering free training. So these videos that you're at today's workshop will have a nice hands-on section at the end. So make sure if you're interested in following along to check the caption for the details. But yeah, we have lots of different resources for people working in phenomics. And if you want to become more involved with PhenomeForce, we have a Slack channel. So you can just email us to be added to the community and get involved. We are always looking for more people.
Yeah, that's true. Also, if you are new here, please subscribe in this video and help us sharing this information with more people, because our idea, as Claire said, is try to help, okay? This season, we have a partnership with Drone to Phenome Group. This is a new community that we are creating for empowering high throughput phenotyping using UAVs in agriculture and plant breeding. As you can see, this is an initiative from different universities, and it's very interesting because in this project, we want to make a survey to discover how people have been using UAVs in their research. And based on this output and answers, we're going to start to make some decisions about pipelines, protocols, and understand how we can improve as a community the use of UAVs. All right.
Mhm.
Oh, I want to use this opportunity also to invite you guys that is watching this workshop for the NEAPPN annual conference. This year is going to be February 22nd to 25. It is in Athens, Georgia. Just type NEAPPN annual conference. This is, to me, one of the most important conferences in phenomics. And as this community is all related to phenomics, I really recommend you being part of that. Claire, Anahit, and I will be there with a workshop, and we are going to bring some people that you already saw here in this YouTube for presenting a new hands-on activity. All right?
Yeah, it should be good. So for today, we're going to have a workshop with Alper Adak, and he's going to be introduced by Seth Murray, and they're going to be talking about UAV pipelines for image data analysis using R tools. So I don't want to take up too much of their time, so I'm going to invite you guys onto the screen. So there's Alper and Seth. And yeah, so we'll hand it over to you guys, and Seth, I'll share your screen.
Perfect.
There we go. Hey, good morning, everybody. And I will also put in a plug for the NAPPN meeting. I really think it is one of the most exciting meetings. Just the community is just awesome and diverse in both every possible discipline you can imagine, and a lot of age distribution and gender distribution. It's really an exciting community.
Right. Thank you, guys. We're gonna be right here. If you need anything, just let me know. If you are watching there having questions, type here in the chat, right, Claire? And we're gonna talk with the speakers. Enjoy. Bye bye.
So Alper is a PhD student with me. He has asked that I give a little context to the work that he's doing, so that you might understand how it fits into a workflow and how it might be used in a breeding program. So I run an applied breeding program here at Texas A&M University. And so I like to just show the slide of corn yields that have occurred based on a historical minimum in 1870 and are scaled based on that in the United States. And I think everybody's familiar with this. The yield of corn has increased 9-fold. And about 50% of that's due to agronomy, but about 50% of that is due to crop improvement. And what we're talking about today is tools that'll really help with crop improvement, but it'll also help with some basic biology and genetics. So what we've done in this time period, we as the scientific community, is move from just using farmers' eyes and breeders' eyes to using technologies that improve the throughput of the breeding process, to be able to screen more individuals and maybe measure them more accurately and measure them in more locations. And so everybody's familiar with genomic tools, I believe, and seed chipping and precision planters. And now we've gone to doubled haploids and genomic prediction in industry. And that's going to lead us into the next big wave of improvement. So one of the things that Alper will be discussing today is data sets that were based off genomes to fields. And for those of you who aren't familiar, genomes to fields is probably the largest public initiative, especially in the corn world, to look at corn varieties grown, the same corn varieties, same corn hybrids grown throughout the United States. And it was started in 2013. We all take similar measurements. So we're down here in central South Texas. And the location that most of this UAV work so far has been collected and analyzed in is here in South and Central Texas. And the data is publicly available on Cyverse if anybody'd like to use it. And then many of these other locations, I think we have 8 locations now that between Natalia De Leon and David Ertl and others, Edgar Spalding, are organizing to collect UAS data. So those data sets will be available publicly, if not already, then soon for anybody to use. And that's what Alper is going to use. I like to make a connection to genetics and genomics, and hopefully it'll make a sense in the next slide. So there's really been these 3 genomic methodological waves. And so in 1986, there was really a focus of QTL linkage mapping began. We were under this impression that we were going to find genes and clone the genes, and that somehow would be useful for crop improvement. What it did do is train a lot of students. It generated a lot of knowledge and a lot of publications, but we just haven't seen the commensurate crop improvement. In 2005, we started seeing the first genome-wide association studies, and it's been sort of the same case. There was a lot of enthusiasm that we were going to find genes that were going to help with crop improvement, but those just haven't, given the investment, haven't translated into the improvements that we hope to see. And then in 2007, we started seeing genomic selection and genome-wide selection with Bernardo and Yu and Mioisen and many other people. And the whole philosophy here is different. So instead of trying to detect some type of biological phenomenon or discover some gene in some type of simple trait, we moved into prediction. So trying to predict the value of each individual in a population, right? And this has been radically adopted by industry. Almost every commercial corn breeder uses some form of genomic selection to help them improve their selection. So now as we think about high throughput field phenotyping, the goal here is going to be to grow more plants or measure the plants better. But so far, most of what you see is automating routine measurements, right? So measuring plant height. So instead of having students with yardsticks go out in the field, we try and do that with a UAV. There's also some estimation of grain yield that's gone on. Most of these are sort of case studies still. They're not routinely being used in most breeding or hardly any breeding programs. We're also seeing disease estimation is really a very popular method to use with some of these high throughput phenotyping tools. But from my perspective as a breeder, what's really going to be more exciting rather than automating routine things is finding new signatures of eliteness. And that's a lot of what Alper is going to be talking about today. Using new methods of measurement that we've never been able to do before, such as temporal growth patterns, which is really a key aspect that Alper has sort of pushed the needle on and is certainly a world leader in understanding how we can use UAV tools in measuring temporal growth. Also spectral signatures that we weren't able to collect on the ground before. And this all leads us into phenomic selection. And I think Alper's published or submitted at least the first study that does a head-to-head comparison of genomic selection to phenomic selection using UAS tools or drone tools. And so that really is an exciting opportunity to look forward to. And then what's really cool about this from the basic biology standpoint and from the applied farmer standpoint is these signatures we find, whether through phenomic selection or signatures eliteness, can be used for farmers to better manage their crop through stress, or it might be useful for identifying new phenotypes of biological importance, right? So instead of letting a growth chamber study dictate what an exciting new trait is, maybe we can discover it in the field. And so this is from a recently submitted article by Alper and a new student to join the lab, Aaron DeSalvio. And so what they actually have been able to do now is automate this routine trait that I never have time to collect in my breeding program, which is both leaf rust and senescence. And so by flying multiple times throughout the year, just in a routine manner, we already had the flight data so that when Aaron started scoring leaf rust or senescence, those algorithms could be applied to data we already collected. And the cool part is we can apply them retrospectively. And I think most of this audience is probably familiar with that. But this is a really exciting tool to add additional traits that could have been collected, but now can be automated. So the temporal aspect is so key here. And so one of the early publications that Alper had in the lab was a culmination of about 7 or 10 years of data and probably 6 different graduate student projects, identifying 2 SNPs from a GWAS study that increased both plant height, but also plant yield. And so we developed some near-isogenic lines. I mean, multiple students contributed to this. And what Alper really, the way Alper really took this over the top was he added this temporal feature. And so when we discovered these height genes in GWAS, in a GWAS study, we only measured height at the terminal level, right, when the plants were tallest. And in these isogenic lines, it's hard to see the difference. I mean, we didn't measure enough plants to actually get significant differences there. But what Alper found was that these differences actually show up very early in growth in the way that we would have predicted based on terminal measurements. And so this has opened up a whole new way of thinking about this trait of plant height, that maybe there's variation that's more observable or more quantifiable early in the season. So then Alper's next paper, and I'm just giving you some background so he can get into the nitty-gritty details and putting this in context for everybody, is that these temporal heights were able to separate our high and low yielding maize hybrids. And we've known that for a while. That's why we started with plant height here in Texas, is that terminal plant height at the end of the season is a very strong predictor for how those plants are going to yield. And in our environment where plants are only 6 feet tall, hybrids are only 6 or 7 feet tall, it's not due to shading. It's probably due to some type of vigor, some type of ability to deal with the heat and drought stress of Texas. And so Alper then was able to use that in a GWAS study to map those loci. Now, Stephen Anderson, a little bit earlier, actually did something really interesting in that he used an algorithm to interpolate plant height throughout growth. So we didn't fly every single day of the growing season, but he was able to actually model plant growth and extract height estimates for every single day in 3 genetic mapping populations. And the really exciting approach, other than just being able to interpolate phenotypes, which is really cool, is to use that in mapping and identify that, for instance, this QTL here happens early in growth, but if we only measured plant height, we would never detect it. So this tells us that there's a lot of really important biology going on through these temporal measurements that have not been captured in the past due to difficulty in measuring them or labor or just infeasibility. So the phenomic selection approach, this is sort of a goofy figure I like to show, is a little bit different. And I don't think everybody gets it. So I'm gonna try and explain this using humans. So this is my family, my parents and my sister. And you can tell that we're all sort of related, right? And this is when I was a kid. And we can tell that we're all related. This is probably 30 years ago at this point. We could tell that we're all related 30 years ago. And this is about 10 years ago at my sister's wedding, and myself and my sister and my 2 parents. And you can also tell that we're related, but we look different, right? And so this relatedness function is what genomic selection is all about. And what we're doing with these UAS tools is quantifying relatedness using phenomic measures. And we can get additional phenomic measures through time. So we can see that we're related here. But if I asked you, how do you know that we're related? What is the indicator? You'd really struggle with telling us, right? But the UAS tools and some extraction algorithms could maybe identify that it's maybe the spacing between our eyes or the size of our nose or the dimples in our chin or whatever, and quantify those differences. And so that's the basis of phenomic selection. So the future of plant breeding, in my opinion, and what Alper has really been working towards just tremendously, extremely productive and outstanding in his anticipation of where we need to go here, is that we can imagine a matrix of dependent traits of interest, maybe yield, but maybe also other things we care about, like nutrition or ecosystem services. And then we have our typical physiological measures we already take. We go in the field and we measure flowering or leaf angle. So these UAS tools aren't necessarily contributing to those. But now we have new temporal phenotypic trait measures. And these temporal phenotypic trait measures are taken over time and are not perfectly correlated so that height at 30 days can predict what height at 90 days is. So these add additional value. These are our temporal measures, right? Then there's additional segregating remote sensing measures, and this is where Alper's work is really going to focus today. And the package that Felipe Matias developed, for instance, has been one of a really critical suite of packages to extract these. So different phenotypes that occur throughout growth. And so those can be put into this prediction. And then we may still continue with some genomic measures, but for my breeding program, those are actually more expensive than phenomic measures. And so that's mainly because of Alper and some of these software packages that allow us to do that. So from all this, we can get relationship matrices, incorporate these in growth models, and then use various statistical analyses. And Alper's really been a leader in exploring different machine learning algorithms for figuring out how to use this data. And functional regression is one that we've worked on with Jose Crosa. He's sort of shared his expertise in that, and that looks really promising too. These can help us predict the best untested hybrids for untested environments, which is what we're trying to do in a breeding program. My last slide really here is a note of caution. For those of you who aren't familiar with the Gartner hype cycle, you can look at our paper, Shi et al. From 2016, looking at UAVs, where we show a figure similar to this. So there's almost always some technological trigger, and then we all get really excited. We say, oh, this is going to solve all our problems. This is going to be amazing. But then over time, we start to get into the trough of disillusionment, right? And so you can think of lots of technologies where we thought biotechnology was going to save the world, and then people, got a little disillusioned. Maybe that's because of public perception, or maybe there's other reasons, right? But then, meanwhile, people keep plugging along. So then there's the slope of enlightenment, and eventually they become productive technologies. So I'd just like to remind everybody that high throughput field-based phenotyping and UAVs in agriculture are probably still in this region. We're probably really excited about them, but we haven't found all the limitations quite yet. Okay, so this is not going to solve all of our problems. We're going to find more errors. A lot of funding that needs to be acknowledged, especially USDA NIFA and the Genomes to Fields project, Iowa Corn Board and Texas Corn Board, for all the work that Alper's done and the various support that Alper's gotten over the years. So Alper, I think this is a great time to kick it over to you. And thank you all for your attention. You're in for a real treat with Alper. Thanks.
Thank you, Dr. Murray. So am I able to share my screen? Okay, that's great. Thank you. So today I will be talking about the three main titles in here. So before going forward, I just would like to introduce my GitHub account, and especially I would like to introduce the material I will be using. So basically, when you enter in here, so I put a README file in my GitHub account. I believe that PhenomeForce crew has already shared this link with all of you. So in order to download the
sorry, Alper, the link to the material you're showing right now is just in the video description below.
Okay, that's great. So anybody who would like to download all the material, so can click link and download all the material in here. In order to download the orthomosaics, there's just one exception. Just click this Figshare link and just click the orthomosaics TIFF file, what we are going to use today. So after this introduction of this GitHub account, I will be talking about what kind of steps are going to be in my code today. So basically, I will be talking about three main parts today. First part is going to be the UAS tool. So basically, the Felipe requested from me to show how to integrate external shapefile into FieldImageR. FieldImageR itself is proposing to create a shapefile to use, but today I will be showing some, optional way to use. Our lab personally is using UAS tool. So also, Dr. Steven Anderson, he is in the audience right now. He created this R package. He will be assisting us when you have the detailed question. But today, as I told you before, I will be showing how to integrate the external shapefile that was produced by UES tool into field image during the reflectance bands extraction. So this is the link if you would like to see the detail. But again, today I will talk about all the steps clearly. So the second part is going to be how to integrate the flight data as a component in our mixed model to deal with temporal data. When I say temporal data, what we are doing in Texas A&M University in the Quantitative Genetics and Maze Breeding Lab, we are flying drones over our population many times across growth, which are, well, basically we are flying drones between 15 to up to 30 times. So that means we can extract various reflected bands belonging to 15 to 30 time points. So in order to assess each single time point as a separate time, we are offering to integrate the flight data as a new component into mixed model. So this part is going to illustrate how to do that in R. And then the last part, what I will be mentioning is the temporal phenomic prediction. We are spending a bunch of time on that to develop this idea, and especially we are collaborating with the really skillful stat person in Texas A&M right now to get the unique idea from them. And then, but right now I will like to show what we have done so far when it comes to temporal phenomic prediction using some machine learning algorithms. So again, before going forward, I would like to introduce some terms, because we are using the range row design in here when it comes to what is range and row. This is pretty well known, but I'm just showing for those who are not familiar with that. So range, basically, this is one of the field, for instance, and in this field, we grown 100 maize hybrids with 2 reps, and also each hybrid was planted 2 consecutive row plot in each rep. So basically, all these lateral block is called range in here, and then so basically each 2 row are representing the 1 pedigree. So when I say range, please consider this picture in your mind, and then that will be easy for all of us to follow the whole step. So now I would like to talk about plant height extraction, but today I will not be showing that just because of the time limitation. I will be focusing on the extraction of the vegetation indices and the reflectance bands, but briefly I would like to show this part. So for each flight data, we are generating the 3D point cloud data and the TIFF file. In here, what we are seeing is just typical 3D point cloud data. We are doing some pre-analysis to do a quality check. For instance, there are a bunch of points at the bottom. We need to remove them. After we remove these outliers from the 3D point cloud data, we are constructing the DSM, DTM, and CHM. So these are very well-known procedure. This is the digital surface model. That means basically that contains the plant and the ground. And then DTM means just ground. When we extract the ground from the DSM, what we are calculating is the CHM, which is also known as canopy height measurement. So this is our plant height data belonging to 1 time point. And then we are merging the polygon. I will be talking about these things, how to create that. And then we are clipping each row plot, as you see in here, based on this polygon. And then we are calculating basically the plant height. So today I will mostly be focusing on the extraction of the vegetation indices. So in here we are using the TIFF file, not the 3D point cloud data. So I'm sure Felipe Matias has already mentioned the detail of the field imager. So basically we are using the TIFF file. We are removing the soil by using this function, field mass function. And then we are creating the shape file by using the UES tool. Again, I will be mentioning all the steps of that. And then after we create the shape file, I am merging these 2 files, aligning these 2 files. And at the end, this is what we are going to get. And when we zoom in, this is how it looks like. So, and then based on this final aligned figure, we are able to calculate the value of the vegetation indices for each plot. And then this is the basically output. So let's jump into the how to use the UAS tool. One of the main functions is going to call plot shape create. So in order to create our shape file for field, by the way, this is going to be our field. As you see, there are a bunch of other fields, but today we are just going to focus this part, the middle part. So we need to create A and B points simply. So A and B point, A basically is going to be point where your first plot starts. B, where your last plot ends. So basically our plot is following like S type. Like this is basically typical breeding planting schemes. And then first two range and the last two range are field. We are not using that. We are just basically using them to protect our material in the middle. So in order to find out the coordinate system of A, so roughly what we need to do is we are using the QGIS external software. QGIS is very well-known software. I'm sure you are most of you are familiar with. So basically, for instance, now I'm going to do slightly back and forth between QGIS and my presentation right now. For instance, this is where my first plot starts. Just click coordinate system, and this is what you need to get. Again, just roughly, you don't have to be exact because we are going to rearrange the shapefile later because of several reasons. And this is what we are going to get the A coordinate. And we need to do the same thing for B coordinate. Once we are done with that, the second step is going to be set the length of the range and the width of the row spacing. And there are some parameters in here. And more importantly, what I would like to mention in here is that this is detailed, but there are two polygons in here you can see. So we can use either of it, but personally, I prefer to use the inner one, which is called the buffer polygon, that is indicated by red color. The one that is indicated with the black color is the plot polygon, which is the outer polygon. So when this is just the figure, when you go to GitHub account of the UAS tool, you can see this figure. But when we look at this figure in reality, this is what we can see. So what we are going to use in this tutorial is going to be the red one. So for instance, there is a row buff in here. This is not visible in here, but when I zoom in, you can see the row buff distance in here. This is just a detail. But the problem in here is that when we create the shapefile, most likely we are going to see these two. This is the raw output of the UAS tool. But in order, well, when we align the shapefile that was output of the UAS tool with your image, this is what you're going to see. So unfortunately, there is a problem in here because each polygon is not covering very well. So in order to fix this problem, after we create the shapefile by using the UAS tool, we need to align the shapefile with our images in, for instance, QGIS. It might be ArcGIS as well, for instance. This is another option. We need to select and drag right to left, and then that is going to increase the, of course, precision in our data extraction. When we finish that, this is the below one is what we possibly going to see. In this case, each polygon covers the two-row plot very accurately. This is what we need to use in our data extraction. This is the first part I will be mentioning in my code. The second part is going to be how to deal with the temporal data. So once we create
Alper?
Yes, for that.
Before you going for the next step, there is a question. Do you want to take it?
Yeah, of course.
Let me share here with you. Hi, Alper. I see you using the SM, the equation, right, for canopy height model to get the canopy height. Can you tell the number of returns? I tried this equation, but our UAV-based LiDAR has only 2 returns, and it didn't not work.
Well, when it comes to returns, I'm not super clear what that means. Maybe the returns can be explained with a greater detail for my understanding. Well, basically returns, okay, I'm assuming that the output and then CHM basically is going to be the number belonging to each row plot, since we use the R polygon, because our polygon, okay, let me go back, for each polygon has the unique ID in it. So we are going to get the number. So Steve maybe would like to contribute.
Yeah, so I think just to clarify, that is the simplistic equation, but converting LiDAR data to a DSM requires 10 or so underlying steps as far as data processing. So you have to filter the points of the LiDAR and then subset them and then convert them over to a DSM and then use that to pull. So there's a lot of underlying steps that are there that we discuss in some of our publications for more detail. I can pull some of those up too.
Yeah, just enjoying this chance here. Thank you, Steve Anderson, to be here. He is the developer of UAS tools, right? So thank you for coming to and help us today in this workshop.
Yeah, thanks, guys. Yeah, I'm just hanging out in the background. Glad to see Alper got to come. I wasn't able to do this on my own, so I'm happy to be here and help discuss some of the work that our lab has done over the last couple of years.
Perfect. Thank you. Here we go.
Okay, to answer the question better, maybe the person who asked this question can have a look at this publication, because we mentioned each step of our plan height extraction in a greater detail. So there are a bunch of steps in here. So maybe the answer can be better when this manuscript is written, especially the material method part. So again, so I am assuming that we extracted all the vegetation. This is belonging to 10 to 15 or 30 time points. So the problem is that how are we going to deal with this temporal data? So basically, there is no straightforward answer for that, but we are offering two-way. So basically, in the first way, we can treat the pedigree nested within flight, basically range, row, and wrap our spatial variation. Since each flight contains our breeding material pedigrees, let's say pedigrees in this case, since each flight contains our pedigrees in it, so we can treat the pedigrees as nested within flight. Or we can follow pedigree-flight interaction. So, and then two-way are basically applicable, but today I will be using the nested design in R coding. So, and then once we created the pedigree nested within flight component, we can calculate the temporal heritability, just because we are going to use the value of the variation belonging to pedigree nested within flight as numerator in here. The other calculation is going to be pretty much same with the normal heritability calculation. And then we had a chance to publish some few our preliminary results. So there are a bunch of examples of these two models in this manuscript if you would like to see the detail. So the last part of my code is going to contain the phenomic prediction. So basically our phenomic data, we are calling it like high throughput phenotyping data, because like we are calling the high throughput genotypic data when it comes to genomic data, when it comes to SMP data, this is pretty much the same. And now we have the phenomic data that contains the various different vegetation indices in it belonging to multiple time points. So so basically we are going to have hundreds columns, changing between 500 to 1,000, for instance, when it comes to our example. So we are using the CAD package simply, and then our phenomic data, we can predict anything we want. So in this case, let's assume that we are going to use the yield, grain yield data belonging to our maize hybrid. So in this case, what we are going to do in the our phenomic data is going to split it into 70% training data, and this 70% training data is going to be used to train the model, like a different model. I will be showing that by using the train function in CAD package, and then we are going to predict the hold on set, like a 30% remaining phenomic data, by using the predict function in CAD. And in this case, what we are offering is phenotypically estimated breeding value. I'm sure many genomic person are watching us, and many people are familiar with the genotypically estimated breeding value, what we are estimating in genomic prediction or selection. In this case, we are offering phenotypically estimated breeding value just because we are using the phenomic data. And then after we predicted the 30% test data, we can calculate the correlation between PBV, which is the phenotypic estimating breeding value, and TBV, which is the true breeding value, the breeding value what we observe in the field. And then the correlation between these two are going to be the merit of our prediction ability. So we are doing these things like thousands times in each training data is going to be randomly selected to see the real performance of our model. So with this, I would like to finish my presentation and start to show the R code. So if you have any question, please write the chat and I can answer about my presentation. Other than that, I would like to start to show some coding in here. Again, before I start, you can download the code in here. These first 4 are going to be the material I will be using, plus the orthomosaics. You can download in this Figshare link. So in here, what you need to do is first you need to install the package, but I just put the dash in here. If you would like to install the DevTools, you can delete the dash and run this code. But since I install this package, I'm just skipping this part. This is I'm opening the library. In order to download install the UES tool, you need to download first UES tool, DevTools, and then you can install the UES tool over the DevTools. So again, you can delete the dash and you can run this code. And then since I did that before, I'm just opening the library in here. So A and B chords, I just would like to recall again so I can go back. These are the A and B chords. So I obtained these coordinate system of A and B from the QGIS simply. So, and then I copy and paste in here. This is what we need first. So, and then I am running these 2 code, and I'm just setting my directory, wherever you would like to save as your output. And this is the main part. So I would like to go back one more time to QGIS to show what are these parameters very briefly. So basically, range spacing, this is the length of the one range, is in our example is 25 feet. So once you set this one, this is one of the important one. Once you set this one, our polygons is going to be with the depth length in terms of height. So, and when we look at this is going to be the range spacing. So all these parameters, what you can see the distance of each parameter in this figure, and then you can set in here. So the out file name, you can just put any name you want, and then your shape file is going to have this name, and also the field we are using the field from 2017, so also you can change the name based on this one. So in here, the important part is that n row plot 2. 2 means in this case, each 2 rows are representing one pedigree. As I showed you before, this is versus I run and then obtain this code before. So each 2 are one pedigree. Just because of this reason, I set the here as 2. And if you set 1, these are going to, like each polygon is going to cover just 1 in here. So when we run this code, the output is going to look like this. It's going to look like this. That means that each polygon is not covering the row plot very accurately. So in order to deal this one, there is some arrangement option for us to do that where we can, click each polygon and drag right, left, up, and down. So I would like to show just one example in here, and then you can do that in your example. For instance, once we run this code, this is what we are going to see. So the buff shape file, this one is the one that refers to this inner shape file. So in order to use this one, you need to use the shape file with buff extension in here. The other one is the outer one. This one is the inner one. And then once we run this code, we are going to get the output I showed you before. But again, this is the arranged one. As you can see in here, for instance, I have already picked this 4 and changed a little bit just because there was an overlap. So in order to do that, in QGIS, I'm showing that in QGIS, just click the toggle editing. There's a pencil in here, and then there is another function called select feature by freehand. Just click and come in the freehand and click this one and select any polygons you want. And after you select that, you can come back in here again. There is a move feature function. Click this one and change a little bit, however you want. So for instance, I change it in this case. Again, first time when you run the US tool, what you're going to get is, there is going to be an overlapping issue slightly, just because there are a bunch of shift can occur in your orthomosaic. So to deal with this issue, what we need to do is we can run open the shape file, and you can open your own TIFF file. You can align them in here, for instance, in QGIS, and you can select the problematic part and you can drag right and left. So this is the important part. Now, after we change, and then you can roughly look at all the other parts. So since I have arranged that before, I will not be spending time on that, but I'm just showing how you can do that. For instance, let's do one more example before proceed going forward. So for instance, let me choose hold this range, and then I can click in here, move the feature, and then click hold the part. And first, let's go a little bit downside. Okay, for instance, this is what we can do. So again, you can choose either one, and you can choose either like just specific range, or you can choose all the area, and this is totally up to you. And I am assuming that okay, I didn't show how to save as. And after you change your, after you rearrange your shapefile, what you can do is just click the pen again, and then basically QGIS is going to ask you, do you want to save as your change? And you can say save. I am going to click disregard just because I have changed before, but in each time you can save your shapefile. I'm assuming that, for instance, you have 16 flights. So maybe the question can be that, do I have to do that for each one? So basically, once you do this rearrangement for specific flight, most likely your shapefile is going to be usable for the other one. So you just need to do these things just for one times. So now I would like to continue. So this part is the part that we are going to create a shapefile. Oh, okay. I forgot to talk about field map. So field map is basically going to be the map of whole this area. And let me to open the field map and introduce what it looks like. So this is the field map. What we used in here. In file should contain your field map. Field map should have the 4-type header. As you can see in here, barcode. Barcode basically is a unique identifier. So we have, okay, how many plots? We have 594 plots in this example. Each contains the 2 pedigree in it. And then these are the plot number, basically the same as with the barcodes, as you see in here, 1 to 594. And then this is the range information. It starts with 3 because our first 2 are filled. Let me open our buffer. And these 2 are our range. Our field. The last one is going to be the field as well. So our first plot starts on the 3th range. So this is why it starts 3 and it goes all the way down. Like, for instance, we have, 20 range in this example. When it comes to row starts with 255. It may start with 1, but for instance, it starts 255 and it jumps to 257. So this is because we have 2 range. We have 2 row plot belonging to 1 pedigree, as I showed you before. This is why it jumps like every 2. Like there are 2 number differences for each row, and it goes from 255 to 319, and it follows the reverse order. So this is the field map, what we need to use. And then let me go back to...
Alper, you do have a question. Do you mind if I ask it now?
Yeah, please.
There's just someone who wanted to know, do the row space, etc. parameters refer to the actual plot measurements on the field?
No. Okay. Let me to go back to slide to answer. Thank you for showing the question.
Perfect.
And this is the just one example of the shapefile. So we have 594 for each one.
Mm-hmm.
So the middle one is going to be our plant material. In this case, we have 2 rows in each row plot. So I guess the specific question is referring row spacing, this part. So the row spacing in our example, when we go back to our shapefile, row spacing is 2.5 feet.
But that is a
Distance.
the distance it would be on the ground.
Yes, we know this is totally belonging to how your tractor and the planting machine are arranged before you start your planting. So these are all the distance we can look at in here to create our shapefile.
I think that answers the question.
I believe. So this is the measurement is totally different things. So we are measuring the plant height with the rules, etc. So okay. I guess, yeah.
Thank you.
Of course. And then this is the code. You can just change row spacing and the buffer, row spacing, row buffer distance, and etc. etc. So that there are 2 line in here. Basically, we are in Texas. for Texas, Texas location is 14 UTM zone with the North Hemisphere. So you just need to change these two based on your location, by the way. So, and then we need the A coordinates, B coordinates, field map, and some arrangement based on the length of your plot and distance of your plot between, row spacing, etc. So this is you need this information to create this shapefile. This is all about it. And then once we run this code, we are good to merge the shapefile with our orthomosaics in QGIS to do a rearrangement. And now, since I rearranged the shapefile, basically this shapefile was rearranged by me previously, where I was sure about that each polygon contains the pedigree very well. And then I can start extraction of the vegetation indices from these images. So in order to extract the vegetation index, these are the library you can install before. Since I have installed that before, I'm just recalling all these R packaging here, but mainly I will be using the field image R. Once you run this library, you can set memory limit size. So I am extracting various vegetation indices and reflectant bands depending on what kind of image I have. So I am always using high number of memory limit, but you can set your memory limit if you want. But I need to do that. So and then this is just directory setting where where the location of your TIFF file is, and then just use a stack function to read your TIFF file. And then since I have done that already, I'm not going to rerun again. And then once you rerun that, there is a function in here called aggregate. So this is important because I would like to spend a little bit of time in here. For instance, if you have the flight that was flown with, like, let's assume that 100-meter elevation. So most likely you don't need to do that. But if you have the TIFF file that was flown, for instance, 25-meter elevation, this is what I have in this tutorial. We need to use the fact equals 4 function. Basically, that means your 4 pixel is going to be turned into 1 pixel. So that allows you to process your TIFF file faster. So this is what that means. If I don't run that, my process time can take even more than days, let's say a couple of days, I don't know, even more. So since I have like 16 to 30 image, so that's a tremendous processing time. So in order to deal with that, this is what I am using for the flights that was flown at 25 meters or below. So in order to give a threshold, let's say this function can be used for the flights with that was flown with the 25 meters. I'm just saying 25 meters for an example, but depending on your computer and the power, this can change. But using more than 4 might be dangerous. So this is why I would like to offer 4 in here for our example, but I don't know which number is the optimal number, but I'm using 4 in general. And then once we get this output, we can read our shapefile that was arranged in QGIS. So let me to show that. Okay, I guess we need to wait slightly. Okay, so this is what we can see in here. So whatever we are seeing in QGIS is also available in here. But the problem in here, we can zoom in specific part of this TIFF file, but personally, I don't like the ability of the R in terms of showing the zooming picture. So this is why we are using the external shapefile, external software like QGIS to zoom in to see the fine detail. For instance, we can see these blanks, and we can't be sure that kind of detail in R. So now I'm sure that this shapefile align with the TIFF file perfectly. So field imager itself proposed its own its own shapefile. You can use that, but when it comes to our plot, we have to be sure that our shapefile should be aligned correctly. So this is why I am using the external shapefile by using ES tool. This is just our example. So I have run this part. It takes a while, like something like 5 to 10 minutes. So this is why I am skipping this part. And I am also sure that Philip has already mentioned the detail of this part. And then what I would like to show in here, this part basically is the part where we are removing the soil from the plant. So let me show one figure related to this. So once we run this code, our shapefile is going to turn from this one to this one. We are not seeing the ground in here, soil in here. So because we removed by using the field mask. So this is this code is basically works for removing the soil. There's a crop value, but this again, I'm sure that Felipe can answer by far better than me. So I'm skipping this part. Basically, this part is this helps you to deal with weed in your field and etc. Hue Index is just the index that helps you to detect the soil and remove the ground soil from your image. So in here, we are defining our vegetation indices. So by the way, this TIFF file, what we are using in here is the RGB one, red, green, blue images. So this is why you're just seeing RGB and some like vegetation indices that were calculated by using the RGB bands. This function is going to propose some well-known vegetation index before. These are BI, GLI, NGRDI, and etc. So you can find these 5 vegetation index in field image R. But when it comes to extraction other vegetation index, so you need to use my index, and you need to define your external vegetation index you would like to extract for each polygon in here. So this is what I have used in our research, and then these are also available in the GitHub account if you would like to use. In order to get the information of this vegetation index, this is where you need to write this ratio in the field imager. You need to basically write the formula of the vegetation index in the my index part in field index function in field imager. So once you did that, again, we are calling the shapefile. This is just the directory where this shapefile is. And once we call this shapefile, we are matching the coordinate system of this output with our shapefile. This is, we need to match the coordinate system by using the projection code in here. Once we did that, we are going to extract the information, vegetation information for each row plot in here, because we are basically combining the vegetation index information. This is the output of this one, and we are calculating the vegetation index information for each polygons in here. This EX1 point shape is our shape file, arrange shape file. So in this part, you need a bunch of memory. So this is why I set the memory in here a little bit high. And then you can visualize our data. And then, well, I'm not running this part because it takes a little bit time. Since I have run this part, you can visualize your data and you can save as the results with like this. So my output is this one. So basically this is there's a time in here. So I just process the one flight time. So this is my output. Since I have run earlier, I just would like to show you today. So, and you can see the example so these are 5 vegetation index in here. These are all other vegetation index I define in the my index part. So I'm closing and going back to our code. Now, since I save as this one, I would like to start reading this again, and then I would like to jump into the second part. So let's read the data belonging to this specific date. And then this part is just I'm putting the name of the vegetation index like as a header, because in the output, let me open that again. In the output, everything was written as like my index 1, 2, 3. Now these are going to have their unique title. So, and then I am defining the name of that. And when we look at the data, okay, let me open my console. So these are basically my data, BI, GLI, and BGI.
We do have another question.
Okay.
So the question is, hi Alper, the vegetation indices you defined under my index, are there definitions, citations also present in any of your publications? My professor will definitely pinch me with this.
Okay, so yes, I mean, these are also very known vegetation index. So, okay, the best answer for that, if you go to, okay, here, if you go to either this publication or this publication, I gave all these, citation belonging to each vegetation index I used in here. So we use the multispectral ones, and even there are more like a citation in here. So anybody can easily find the citation when they visit this manuscript.
Perfect. Okay, thank you.
Mm-hmm. So now we define the title for this data, and now we are good to go. Again, this is just for one time point, which is this one. Now there is another field map. In this field map, let me show you field map 2. In this field map, we are having the pedigree information, range, row, rep information. These are just flowering times and the plant height. These are the yield. But the important part is that we need the pedigree, range, row, and rep information to run our mix model. So in order to do that, but there is no index information in here. What I would like to do is I would like to merge these two files in R. So this is the first 6 row of the field map, and this is our data, and we are just going to use very well-known package duplicate in R, and we are going to use a merge function, and ID column, the unique identifier is common in field map and the data. So we can easily merge, and we can look at that. Now I merge our field map with the vegetation index. So basically there are a bunch of vegetation index in here. We have this is the merge part coming from the field map in here. So we have the pedigree information, range row wrap information, and the other hand measurement trait. These are like flowering times and the plant height, and then yield. So now we are good to proceed to run our mix model. So we are going to use LMER basically, and then once we call the LMER package in here, now in this illustration, I will show the mix model just for single time points. So in this case, range, row, rep, and pedigree should be factored. And then again, this is our data. And simply let's pick up one of the vegetation index to run in the mix model. So I just pick randomly GLI. So you can use any vegetation index to run your ANOVA. So let's run the GLI ANOVA is going to be my output just because I am predicting the GLI in here. And when we look at the results, this is what we get. So let me calculate the percent. So basically what I did in here is that I just calculate the explained percent variation in terms of explained variation as percent. So now I add this column, like we see basically variance component, and then we can look at how much explained percent variation belongs to which component in here. So basically, for instance, pedigree explained the 20% variation for a GLI index belonging to one time point, by the way. So, and then we can calculate the heritability, like basically 20% divided by 20% plus error divided by 2. 2 means we have the 2 rep. So this is typical heritability calculation. And then when we calculate the heritability, for instance, for this specific vegetation index, which is GLI, we calculated almost 0.8 heritability, which is like this trait looks like very heritable, this vegetation index. So in order to calculate the bull-ups, because we treat the pedigree in this case as a random effect just because we put the 1 in here. If you remove that, you can treat your pedigree as fixed effect. This is totally up to you and up to your research. And there are 2 ways to calculate that. You can either use the random effect, but you need to add your intercept on it to calculate the breeding volume. So now we can see that, okay, these are our pedigrees, and then these are our breeding values. Or you can directly use coef function to get the same value without adding the intercept on it, because coef function contains the intercept in it. So, and then we can look at the histogram. Okay, I guess this should change. Let me do it again. So anyway, I can just show the there is a slight problem in here. Let me look at that.
Eu acho que tá Oh, I start speaking Portuguese. I guess it's missing one. What the name of those guys?
Okay, so I can reopen this part. Well, it's okay. So I guess like there's a some...
You know, exactly. It's missing one. You need to close the. What's the name of these?
Bracket. You need to close your bracket.
Now I can fix. So let me clean this part. I guess this is the reason why doing that.
Yeah, on the line 179, you need to close the bracket. Yeah, that's 179. Yeah.
179.
179 in histogram function.
Mm-hmm.
You need to put at the end. Yes, close it.
Okay.
Here I go.
Okay. Because,
yeah, perfect.
Very, very annoying.
Oh, that's always happened with me. You have no idea.
Okay. Yeah, I mean, let me to read this data one more time, and then it should.
Sure.
Fine. Here we go. Okay, thank you, Felipe, by the way. So we just forgot this one parenthesis in there. So, and then this is the runF function and the core function, and then we can easily extract the breeding values, which is in this case best linear ambiance predictors, not blues, and then we can calculate the blobs for pedigree, and we can simply show the histogram on the right side. This is the blob values of the pedigrees for GLI index. Now, I would like to illustrate the how to run the mixed model for multiple time points based data. So in order, before going forward, I would like to introduce this file. And let me to open this file to show how it looks like. So again, this is all about our like a planting times, planting like information. I'm sorry. This is the barcode name, and this is the year. This is the test data. We can easily disregard these things. What is the important part is the plot, rep, pedigree. These are all flowering times and the plant height data. I'm skipping this part all the way down. Range row is another important parameters we are going to use in our mixed model, because these are basically a spatial variation. And then I calculated all of these vegetation index for each date. So if you ask how many date I have in this file, for instance, I just show the 1 day for you, but I did the same analysis, I did the same extraction for multiple time points. So in this case, there are 15 flight dates in here. So basically, these numbers are showing days after planting time. For instance, this one shows for this pedigree, basically this is hybrid, and this is the value of this specific hybrid belonging to 20 days after planting time. So, and then these other values are belonging to this time point for the first pedigree. And this is the second pedigree, and it goes all the way down. So basically, this is a pretty much big data for us. Now, I would like to integrate this column as a new component in our model.
Before you move on, we do have one question.
Mm-hmm.
Hi, Alper. Could you please show me again where did you take from which output the intercept to calculate your BVs?
Oh, okay, sure. Basically, I passed this part slightly fast, I guess. Okay, so I guess the question is, once we run the LMER, pedigree is our plant material, range row wrap is our spatial variation, and when we visualize the GLI ANOVA, this is the intercept in here. This is where it comes.
Okay.
So basically, this is the answer. So we can add this intercept on the run F results, but there's an easier way. You can directly call coef function. This one contains this both information at the same time, but this is totally up to however you would like to run. There are both options I'm showing in here. So I guess this is the answer, hopefully.
All right, thank you.
Okay, of course. And so I was introducing the CSV file containing the vegetation. This is belonging to multiple time point. My first example was for the one single time point. Now this is for multiple time point. So in order to integrate this column as a new component in our mix model, so as I showed you before, I guess better to go back this one. I will use the nested design here because flight is going to be the new component, like basically this column, days after planting time, is going to be our flight component. And pedigree are going to be treated as nested within flight. Range row wrap also are going to be nested within flight because each flight contains our field and of course our pedigrees in it. So this is why we can treat the pedigree nested within flight. But I would like to also mention that, so this is a technical detail, but I guess this here is a good place to discuss that. So there's a violation in for nested design because, for instance, let's consider the genotype environment interaction plus rep nested within flight. So I'm sorry, rep nested within environment. So this is pretty well-known model in plant breeding to dissect the G by E very simply. So in this case, we can treat rep nested within environment just because rep are not same in each environment. But in this case, our row plots, our experimental design is going to be same from flight to flight. That's a violation. Maybe we can't use the nested design. However, the plant are not going to be same from flight to flight. Plant are going to keep growing. So the appearance of plant are going to change. So basically, even though the location of the row plot is going to be same from flight to flight, the condition of plant as well as the environmental factor are going to be different from flight to flight. Also, this situation allows us to run the nested design. So I didn't know the answer. Answer looks like finding the black cat in the dark room, but again, there are two options. These can be used. So again, I will be showing the nested design today. So let me to continue to run the ANOVA mix model for multiple time points. So I read the file I showed you. Okay, I can't read because, okay. This is the directory again, and then I read the file and I look at the structure of the file. Okay, this is, and then what I need is I need the pedigree range and row. Range and row looks like integer in here. So in order to make them like a factor, so I need to make the component I will be using in my model make the factor in here by following these codes, like as usual. And then I would like to show how it looks like first. Okay, I need to open the, by the way, we are going to use the ggplot right now because I will be doing some illustration for temporal data. So this is basically how it looks like. So x-axis are showing the days after planting time. Our flight time is days after planting. And the y-axis, I'm sorry, the y-axis are showing the value of the GLA index. So these are basically the value of the pedigrees belonging to the specific time point, flight times, drone times, and then drone flight times. And then y-axis are the GLI index, green leaf index. But this is the raw data. I didn't run the ANOVA right now. That contains the information of both rep. And now this is the code. In here, that basically is flight time, as I showed you before. This is the column I will be using as a new component along with range row and rep and pedigree. So okay, I guess this is like better. And then as a predicted variable, again, I just choose, I guess, GLI in here. And you can practice by choosing any of the vegetation indices you want, like for instance, NDRBI, NGBI, or anything you want. So let's run the ANOVA for, again, we are using the GLI in here. Our output is going, our data frame is, our data is called DF in here. We need to define the DF in here. And then all this component is basically coming from this design. I will run that. So it's running. So now it finished. But if you run the interaction model or this model, pedigree flight, pedigree nested within pedigree flight interaction, it takes slightly like more time than pedigree design just because of the calculation. And then let's look at the results. So this is our intercept again. It shows in here, but let me to calculate the percentage of the variation. So this is basically like easier to me to follow. So basically these are the percentage of the variation explained by the component. So for instance, 78.9%, almost 88%, the total variation explained by flight DAP. So basically 1, 2, 3, 4, 5, it follows like from left to right in here. And then you can see that basically the highest explained percent variation is coming from the flight data. So you can see the differences in here as well, based on, like I'm talking about visually speaking. But when we run the mixed model, we can easily say that highest amount of explained percent variation is coming from the DAP, the AP, which is the flight date. And so this is the some coding in here we can easily follow, but all of these things is just for calculating the showing the value for, well, what I did in here is that very simply I just assigned the title and then I calculated the interaction in here. Again, I add the B in this case. In here, B is the intercept, and then this B was added on the pedigree value on it, and then I just gave the title, which is the T blob T block means temporal breeding volume for GLA index. And then I separated the column. Basically, column seems like that first. Like this is the pedigree information. And then there is a sign in here. And then this is the days after planting time. In order to separate that, I just follow this 2 code. And then when we open it, so we can, like, it was like that. And then I separate like as days after planting and the pedigree in here. This is some, separation I did in here. So in order to calculate the blob belonging to pedigree and nested within flight components. So basically that is the flight in here. That is the abbreviation of days after planting. And in order to calculate the blob belonging to this one, I need the information of, I need the breeding volume belonging to that, plus I need information of the pedigree itself. So basically I need to sum the information of these two to calculate the pedigree breeding volume belonging to specific time point. So this loop does that. And then when you use the str split to split this column, interaction column, into DAP and pedigree, your results is going to be the list function. So in order to get rid of list, so you just need to use the unlist function to make your list data as a data frame. And then what I'm doing in here is that now I calculated the temporal breeding volume belonging to each pedigree. Temporal breeding volume means like the breeding volume of each pedigree in terms of GLA index belonging to all this flight time. This is why we are calling temporal, since we are using multiple time points. And then I am sorting the flights from, like a descending or ascending order. And then when I visualize the data, it takes a while. This is what we can see. Basically, each line in here is the one pedigree, and x-axis are the days after planting again. I was showing the raw data to you. Now this one is the temporal breeding volume. Well, we are calling that as temporal breeding volume, but again, temporal breeding volume in this case, the breeding volume belongs to multiple time points. This is why we're calling it as temporal. And each line, as I told you before, represents a one pedigree, and we were using 280 pedigree for this specific example. So you can visually see that how the GLI score change across time point for specific like hybrid. So now this is just for GLI. So we have multiple vegetation index we extracted, if you will recall. Now I run this nested model for all vegetation index, and let's see how they looks like. So this is G2FE_2017 tblob file contains the results of this model for all vegetation index. So I was showing just GLI in here, but this one contains all of them. So let's see how it looks like. Now this is my directory again, and then I am reading the file that contains all the vegetation index, tblobs of vegetation index, and then let's look at that. So these are basically, this is the huge file. Okay, maybe it's better to open in here. I just read this file. Now I have a bunch of columns. Let's see how many columns. 540, for instance. So let's look at the BCC in here. What that means is BCC belonging to 103 days after planting. BCC belonging to 105 days after planting, up to BCC belonging to 9 97 days after planting. There are should be 16, I believe. Yeah, 15 time points in here. So basically, I have the vegetation index belonging to 15 time points. So this is our data. So these are DTA and DTS, day stand thesis and seeking, belonging to maize hybrid we collected in the field, and plant height we measured with the stake, ruler stake, and then GY is the grain yield. We are going to predict them by using all these phenomic information at the end. But I'm just showing how we should read the headers in here. PCC is the vegetation index, and the number shows which days after planting time information we have for what kind of vegetation index. So basically, this is vegetation index, this is days after planting time. And this is the pedigree information in here. These are our pedigrees. These are our maize hybrids. So we have, okay, we can look at that. Okay, we have 280 maize hybrid in here. This is our phenomic data, high throughput phenomic data. And the only differences in here is that, like, think about any S&P data, it contains zeros and ones and twos, and these are basically categorical variable, but we are treating them as continuous variable to use in, for instance, RR block or any prediction model. But in here, these are naturally continuous variable. So we are considering this situation a lot just because the number what we are using in here, which are phenomic data, is also the continuous data. So let's assume that we read this data in our, okay, in here. But before going forward, there is a really common question I received recently when I presented that in various conference, and one of the questions was that what if these vegetation indices are correlating between one another, referring the multicollinearity problem. So yes, all the vegetation indices were calculated based on tree bands. However, just because we have 15 time points, multicollinearity problem was inhibited by the temporal dimension. So let me to calculate the correlation of the phenomic data, and then I would like to illustrate it as a matrix, heat map matrix, to see what kind of correlation we are going to get, and then you can do your own, biases about multicollinearity problem if there is. So now we need this package to create the heat map. So correlation, we are calculating correlation, but we are calculating correlation from 6th column to all the way down. That means from here, because the 6th column is the starting time of the vegetation index, and it goes all the way down. So now I am calculating the correlations for phenomic data. This is just for assessment of the multicollinearity problem. And then just color palette, I am going to use navy, from navy to white, white to red. And let me make the heat map. So it takes slightly time. Yes. For instance, now this is the correlation matrix illustration of the phenomic data. So the navy color refers -1, 0 correlation is represented by the whitish color, and the 1 basically show the high correlation. So the correlation are changing across time point and across vegetation index. So again, like telling there is a multicollinearity problem or not, I guess that one of the best ways is to calculate the correlation for the your phenomic data. This is what we can show in here. So the correlation are changing. So there is same correlation for the GBS data. When I calculate the GBS data, this is there is the same scenario. So the GBS data is correlating between like SNPs belonging to specific chromosome regions are correlating as well. But these data has been using in genomic studies in the last 15 to 20 years. So the same scenario for phenomic data. So what we are seeing in here is that like pretty much the same scenario. So like saying there is a multicollinearity problem or there is no multicollinearity problem in the phenomic data is, you know, I don't know the answer, but this is what it looks like. Like visually speaking, the correlation is changing between -1 and 1. So I am continuing. Now I would like to visualize the phenomic data to see all vegetation disease across all time point. And I also would like to use the yield data to see the separation. If in this case, separation means maybe the value of the vegetation index has an ability to separate high yielding and the low yielding hybrids. In order to do that, we are going to use reshape. We are going to stack the data. This is the white format. This is white format. I would like to see the vegetation index in one column and the days after planting time in one column. So in order to do that, we are using the reshape and the math function. And then these are what I don't want to stack because these are our hand measurement and the pedigree information. And then I stack the data. So the white format basically became something like that: the flight data and the vegetation index and their values. And then, since I use a str split, this is using the list function. In order to get rid of list, we need to use the reply unlist function in here. And then now this is our data. We have the pedigrees as a character, and then we have the value of the vegetation index as numeric, and the flight as character, and the vegetation index as character. But what we need is, I would like to use the flight data as continuous because growth is follows a continuous pattern. So I would like to make the flight as numeric. So in order to do that, I'm just going to use this function. Oops. And now this is a little bit huge ggplot code in here very briefly. Okay, let me make that below. So this is the stack data. Flight is going to be on the y-axis, and the value of the vegetation is going to be the the flight is going to be the x-axis, and then value of the vegetation, this is belonging to each time point, is going to be on the y-axis, and we are going to make grouping by using the pedigree, and we are going to use the GY. This is the grain yield for making the color for high yielding and the low yielding hybrids, but it's going to be continuous, not categorical. And then this is the geom line. We are going to use the line, and alpha is like basically a shading factor, and then the size is, since we are going to see a bunch of line, I want to use a little bit like not thick lines. Face wrap, vegetation indices. So I would like to separate all vegetation indices as a different section, and then I would like to see 6 column from left to right. So these are all others totally, like when you change these ones like numbers and the colors, you can change the appearance of the ggplot. But what I would like to show in here is that since we use the flight in here as a continuous, we can set the days after planting time from low to high. So let me run that, and when we see the output, everything is going to be more clear for us. It takes slightly while. Okay, now this is just this is the grain yield starting from 5 to goes up to 13 something. This is ton per hectare unit. But the the situation is not clear in here. So in order to do that, I would like to use the navy color for low yielding ones and the red color for high yielding ones. And the mid value is going to be the white for the hybrids with these values. So now I would like to, so midpoint is 10.5 because I know I calculated that the average of the grain yield is 10.5. Now I would like to do some coloring. And I'm doing these things because I would like to show that vegetation index, temporal information of vegetation index has the ability to separate high yielding and low yielding hybrids. Let me to zoom in that. Okay, we need to wait slightly, I guess. Yes, first, this is example. So these are all the vegetation. This is I run before, but what we run is where's the GLI? So this one in this tutorial. But again, you can practice as much as how you want for the others. So for instance, low yielding ones is the navy color, the high yielding one is the red color, and the mid yielding one is the basically white one. So let's pick again GLI, for instance. GLI index in here, for instance, follows. For instance, this is the highest, the hybrids with highest yielding, the hybrid with the highest yielding. So it follows always higher value in terms of GLI, like not always, but in general, highest value across the time point. Time points, the flight times are visible on the x-axis, and GLI score are visible on the y-axis. I'm sorry, the value of the vegetation indices are visible on the y-axis, which is specific to each vegetation index. And then we can easily see that the hybrids with moderate yield value always follows in the middle. So the hybrids with the higher yielding values always follows the highest value in terms of GLI index, and the lowest one is following really low temporal value of the GLI. This is how we can read for GLI, and you can look at the same. Well, there are the same scenario which is applicable for many other vegetation index. So that means temporal vegetation index has an ability to separate the high yielding and low yielding hybrids. So did I pass the time?
Yeah, no, actually, we just came to tell you that that's very cool. I guess when you give a more biological application from the data that we are extracting, that's what we really need, And I'm also doing something very, very similar, and I'm glad that to see that other groups also are going the same direction. Yeah, I'm glad to see this.
Thank you so much. So that's a nice break to me, and at least I sipped one water. So I'm very close to finish, by the way. So I know it's pretty much boring, but
no, it's been cool. Yeah, you have around 10 to 15 minutes.
Okay,
right,
so 10 to 15 minutes is going to
be got time left.
Okay, thank you so much for warning. And before I close this part, this is the variable. Variable means this is the canopy height measurement. I didn't show how to extract the canopy height measurement, but what I am showing here is that we fit the temporal plant height data by using some functional parameters, which is called variable. Variable in this case, like a S-type growth, basically, very basically. And then we publish that. If you look at our Google Scholar account, there are some examples for variable fit. And then basically the temporal plant height data are following some ups and downs at the end. So basically the plants dry out and its plant height reduced for some reason because of the biological reasons, of course, of senescence, and because of the wind, some plants lay down or lodge. So in order to inhibit that, so we are using the variable fit. So this is all about it. So I don't know what I can tell more about that, but these are the vegetation index we extracted for multiple time points, and we visualized it by using the yield value, of the hybrids, and then we can easily see that high yielding, low yielding, and the moderate yielding hybrids and their temporal pattern for different vegetation index. Now I'm done with this one, but if you would like to, for instance, calculate the correlation for specific vegetation index, now I would like to do that, because this is one of the another question that was asked to me, like the correlation between vegetation index and yield. And then I will be talking about the temporal phenomic prediction, and I will conclude my tutorial today. So in order to do that, again, I will be using the T-blob white, like I have already read that previously. And then I will just choosing NGRDI, like that means I would like to pick any column containing NGRDI, because I'm using the contains function in select function. And then I would like to call the GY. I would like to have the GY, and I would like to see the correlation of the temporal NGRDI value with grain yield. So I would like to be a little bit fast. So these are the package, what we need to calculate. So I am calculating the correlation between NGRDI, temporal NGRDI value, and the grain yield. And then this is the PMAT means we are calculating the p-value for each correlation. And then I would like to show that. Okay, for instance, this one is showing the correlation between grain yield, GY is the grain yield, and the NGRDI score belonging to multiple time point. I would like to show that for the grain yield is correlating with the NGRDI score belonging to 63 days after planting with 0.6 value. So which is like, to me is high, is very nice and promising. Also, there are some promising, like it's changing between 0.5 to 0.6 in general. The correlation between grain yield and NGRDI, temporal NGRDI score changing between 0.5 to 0.6. Also, it reduces between, for instance, the lowest value, I guess, is NGRDI score belonging to 41 days after planting time, because the correlation is 0.27 with grain yield. So this is what we can do for how we can calculate the correlation between grain yield and temporal vegetation index, like any certain index you want. In this case, I just pick NGRDI, but if you change the NGRDI with other vegetation index you would like to see the correlation between specific vegetation index and grain yield, you can just change this name with whatever vegetation index you would like to pick. And then this is the phenomic prediction model. So, basically, if you ask what that cross means in this case, this is the correlation which is not statistically significant based on 0.05 I set in here. So this is the last part. I would like to show some phenomic prediction here. Okay, there's a typo in here. Let me correct that. So this is all the library you need. So if you ask what are these, this is the CAD package we're going to use for prediction. PhenomeForce is a package that you can input your data. So unfortunately, current package doesn't tolerate the missing value. So you may want to input your missing values by using the PhenomeForce package. I don't have any missing value in this phenomic data, but just in case, I'm putting in here. And then this is how you can set your core, how many cores you would like to use. So I have 16 core in my computer. So that means I would like to put one core for to keep your computer alive. So other 15 core are going to be used during the prediction. And then let me to show our data, what we are going to use in our prediction. So we are going to predict grain yield, and we are going to use 540 columns to predict grain yield. And then before going forward, you need to use PhenomeForce package that allows you to predict your your data if you have a missing value. But I can run that. Okay. So, and then I am defining, by the way, I will delete first 4, 1, 2, 3, and 4. And I just would like to have grain yield and all the vegetation index. So this is why I'm running this code. So my output in this case is going to be 811. And then when I visualize 811, this is what we can see. This is grain yield, and this is the BC, all the vegetation indexes from left to all the way down, all the way right. So, and then I can, input my data if I have the missing value. So since I don't have, it basically didn't take time. And here we can set the cross validation. So basically, for example, I'm using 5-fold cross validation with 3 repeat. And then this is loop. So I am going to use just 5 loop, but what we need to do is at least we need to use a loop between 1,000 to 10,000 to assess the real prediction ability of any model you would like to use. So I put a bunch of example in here. For instance, there are how many? Okay, this is LM means linear model. So first, we need to create the external empty matrix to fill our prediction. As I showed you before, our prediction is going to be this scenario. We are following this scenario. Like 70% is going to be used to train model, and 30% is going to be what we would like to predict, which is the test data, validation data. And then we are going to calculate the correlation between estimated breeding value of the test data and the true breeding value of the test data. So now I would like to run all these empty matrices, and then these are the variable importance scores. These are the output of any machine learning model to see which vegetation index belong to which time point is most important. For instance, you can get that. And this is the tuning parameters of your machine learning model. And this is the bootstrap. So in this case, we are doing the data partitioning in here. P0.7 means I would like to take the 70% of data as training. And then the E21 is what we are going to use as data. And GY is what we would like to predict. And then since I am going to use just 5 bootstrap as an example today, this is what we can see. AC LM1, so for instance, LM1 in this case, we are predicting training data, but LM2 is predicting LM1 is the one of each model, means that we predicted the training data, and then we calculated the value of the training data with predicted data. We are calculating correlation between actual value of training data and predicted data. And the 2 is the same for test data. So 2 is more valuable. And then when I run this model, when I run this loop, so basically, okay, there's a problem because, okay, I didn't define the bootstrap before. Okay, now my code is running. So in this case, basically, each model right now is being run by using the same training data in each bootstrap. So, and then you can calculate the second of the number 2 means the correlation between actual value of test data and the predicted data. Now my code is running, and also this is the end of my presentation. All the code is available in in my GitHub account. If you have any question, just feel free to shoot me an email, and I will be happy. So I guess time is over for me. So I will be happy to have your question if you have. Thank you. The last part was a little bit fast, but again, this is totally related with the machine learning stuff. But I would be happy to show anything when somebody have a question or concerns about these coding.
Yeah, thanks, man. That was great. Yeah, you did a great job. Yeah, I'm telling you, I'm really doing something very similar, and I'm very happy to see you're doing as well.
That's great. So I guess there was a question I saw. A question, not really want to contact your codes and libraries are so cool. Oh, thank you. I have been learning R for years, but I have never used some of your packages before. How do you learn and perfect and R skills? I'm not definitely perfect. I'm sure Felipe is more perfect than me. There's no perfect when it comes to R, I guess. Like from my true knowledge, every day I'm learning something. Yes, this is the best answer, I guess. I know. I mean, please, you guys tell your own experience, but I've just learned everything from, online and some courses and etc. I don't know the answer, to be honest, but I know that I'm not perfect. I'm still learning.
It is doing, right? So every day we learn
Yeah.
a little bit more.
Yeah, it's a lot easier to learn on a real example than necessarily following someone else's tutorial when you get into it with your own data.
Yeah. So that's all I get.
I do have some questions that I took some notes here. So please, guys, if you are over there, send questions here. And for Alper or for Steve or Seth Murray, also we can invite Steve and Seth Murray if they want to come in. Well, the first question is, did you make any kind of flight curation? For example, you evaluate some CVs for coefficient of variation for flights, and then, for example, the flight on the 60 days not was great or good, and maybe that's going to put more error or bias in your analysis, then you remove that. If you did, what type of curation you use it?
So that's a great question. Basically, we have the technician. He is the UAV pilot. Basically, he's flying the drones like a different time, depending on environmental conditions and etc. etc. So all these environmental conditions, even the different times of a day, can cause a problem for orthomosaics. Well, during my tutorial, I told that we are flying drones from 16 to 30, but we are not able to use 30 of them, all of them. So of course, some of them contains hole in it, for instance, like basically black holes, and we can't use these orthomosaics. This is one of the major problems. First, we are eliminating these orthomosaics. Second one is the resolution issue. For instance, one resolution is not matching. All the resolution are not matching from one flight to other, belonging to a specific population what you are interested in. So also we are deleting that. But your question is mostly related to CV values, correlation coefficient values, when it comes to which flight data should be used or which one should be eliminated. So I haven't used the CV, but it sounds like a good way to decide which flight should be used or not. But mostly I am eliminating the flights based on its appearance and the quality, to be honest. I'm not doing some statistic test to decide which flight should be used or not. But overall, temporal data has lots of ups and downs, as I showed you before, depending on, for instance, flowering time when the tessel appears for like maize. and then we are seeing some ups and downs. But we can, smooth this part by using spline methods. I didn't show in here, but I showed basically variable fit. We can fit the temporal plant height to, tune the little ups and downs occurring at the end of the growth season for plant height. But right now, for instance, I didn't show, but I can at least tell verbally. We use the Gaussian fit, which is kind of a kern belt fit. We can use that for vegetation index to deal with, some bad flights, and we can use that instead of removing them. I guess I can answer like that.
Nice. On the QGIS, when you were showing the QGIS in your presentation, your shapefile or your grid file, like each polygon is separate from each other, right?
Yes, each polygon has, I can show like that. For instance, if I go to any polygons and if I put my cursor on it, I can see the name of, this is the 12th. This is coming from the field map. For instance, I am dragging to right one. This is going to be 13. This is going to be 14. And I am going to get the value of vegetation index for this specific plot just because of this unique ID.
Yeah, perfect.
And also there's a really nice example because there's a really nice variation in here. For instance, this one senescence earlier, the other one. So for instance, we can see that. I know what kind of genetic background is there in here, but I'm just showing you one example. So even we can score the senescence. So I guess Dr. Setmuri showed that in his slide. For instance, let's say that it looks green, right? Let's put 5 for that. This is senescence. Okay, 5 is complete senescence, for instance. We can put 5 for that. We can put 3 for that. We scored the senescence by using the last 4 fly, and we predicted the senescence to understand which vegetation indices are playing a major role on prediction the senescence score by just sitting in my chair and look at these polygons, and I can quantify the senescence. I know I'm a little bit off your question, but go ahead.
Actually, this is the one of the perfect examples where there is no perfect tool for do anything, right? And then this combination where you go to R, come to QGIS, which is normally Python-based softwares, and also use different R tools like UAS tools to do one step. That's exactly what John to Phenom group are looking for. This type of pipelines where people combine tools to get a final result. Yeah, good job, dude.
Okay, thank you, Philip.
Claire, do you have more questions?
I have a couple of questions, but they're not big ones, and we are over time. What do you think?
Yeah, maybe if you do one last question, and then we say thank you for Alper and the other members here.
That sounds good. Well, my question just kind of relates. I don't know if you saw this comment when it came in. Someone was just saying that they're from Africa and they really appreciate that you were showing stuff using RGB data because those are more affordable sensors for them. So do you think that that you're obviously you don't necessarily have the same orthomosaic steps at the beginning if you just have an RGB camera? Like for the platform that I use, we go between our plots and take pictures. Do you think that I could use my RGB pictures in the same kind of way as you are and apply maybe your downstream analyses to them?
So yes, if you are planning to take your plots with your own any cameras which is really close to your plot. So as I told you before, this picture, what we are seeing right now was taken with the elevation of 25 meters. So in this case, I'm assuming that your distance is going to be even less than half meter. So, and then when you merge this file, basically your TIFF file is going to be like a 10 gigs, I believe, gigabytes. So in order to deal with that, I am going back to my R to show something really quick. There's a fact function in here. You need to use that. here. Maybe you can use this effect as like 20 or 30, I don't know, to reduce the resolution. By the way, in here, we have like, there are a bunch of pixels in here. I'm zooming in, so this one is 1 centimeter. Each one is 1 centimeter pixel resolution. So your resolution is going to be by far bigger than this one, just because you take the picture very close.
Yeah.
So yes, you need to do some downstream analysis, such as you need to use fact function like a little bit higher maybe than before.
For sure. And there was one more question that just came in. Concerning plant indices data extraction, can I extract per pixel of each plot with field image R?
Well, basically, I answered this question. So this is totally up to how powerful computer you have. Like we just recently bought a computer. Yeah, Dr. Setmuri bought a really nice computer to our lab. Now we're using them a lot. And sometimes what I'm doing is I'm clicking the enter, and then I'm going back to my home and going back to school again, and then I am getting the results. So this is how I am doing. And roughly I have 16 and 32 cores in my computer. So this is a lot. And then I have really huge, great screen card, like GeForce 2060, and then also Quadro series. We have Quadro series, and I have 2 screen cards in it. So, and even though I have this qualification, I have to do fact equals 4. So when it comes to extraction, the vegetation is per pixel. Well, if you run this code without that, it takes, I don't know, I don't want to exaggerate, but days. It takes days.
I do the same. I support your your method. Yeah, it's the only way. It's because at the end we are just taking the average per plot, right? So the result is very the same. I did the comparison actually before, and then the result, the correlation, the results is like 98%.
Yeah, 98. Yes. We are not losing anything. Well, what we are interested in when it comes to plant biology and breeding, we are interested in the heritability. So if the heritability is good for us, we are good to go. So now I showed that heritability is 0.8 belonging to specific time point. So then we are happy.
We did have a question that I think would be good to ask now that Seth has rejoined us. Someone was just curious about the manpower you have in your lab to accomplish the work that you have, because obviously there's a lot of steps before even the kind of stuff that you're doing downstream in terms of actually analyzing the data and getting to outputs. So can you talk a little bit about your lab?
The question is nice. But Dr. Setmuri should answer that.
Yeah, I mean, so the way we set it up at Texas A&M, and then what I did in my lab is there's one person whose focus is just collecting data. They fly the drones. Scott Wild, he flies the drones and then assembles the orthomosaics. And then, part of his role is making that data available, for instance, on Cyverse, where some of it is. But I mean, he's not done with all that from this year. That's his role as he collects data. And so what we set it up as is, instead of focusing on a single trait and flying just to answer a single question, it's really a platform where we're collecting data throughout the season. And so regardless of what questions come up or what you want to try and apply, there's hopefully a flight that matches up to it, right? So it's a little different of an approach. So that's one person. And then everything else that you saw today was done either by Stephen developing that package or his paper, or by Alper. And Alper works with some other students here, but I think everything that he showed, he did the data analysis and the programming and the paper writing. So 2 people for most of what you saw today, I guess. And then I just talk about it and brag about them. That's my role. But before Alper jumps in, there was a question earlier that I think is really important about the RGB. And so, when we started our UAV project here at Texas A&M, there was a big interest in, especially among like the ag engineers, they wanted to always collect hyperspectral data because that was new and somebody's already published on RGB. And we were told often that, plant height would be easy. And 5 years later, we're starting to get a handle on plant height with RGB, right? And so it was very intentional that I have not invested in really fancy drones, because we're getting so much more information from this temporal aspect. And when we had a Ugandan scholar here who was doing some work on perennial sorghum. I mean, if she was to go back to Uganda, she can get the same drone and fly it there and do the same work. And if we start focusing on hyperspectral, you know, it's just not scalable to every breeder's program, right? I mean, people invested a lot in genomics tools. They're not going to be able to stop all that and then jump over and all buy hyperspectral cameras. So it was intentional.
Awesome. Yeah, we are also in our group. We are also like holding the flag for the open source and also low cost phenotyping platforms where everyone can do it from where they are. Right. So we are in time. Thank you so much, guys. Thank you, Seth. Thank you, Alper. You did a great job. And every time when you guys have a new pipeline or a new tool, let us know, and then we can set a workshop here and share this with our community. Okay?
Of course. Thank you for inviting us, by the way.
Thank you all.
Bye-bye. Thank you.
Bye-bye.
So that was our last workshop of the series, right, Felipe?
Yes, so finishing here till 2021. So thank you, Claire. Thank you, Anahita, that she's not here today. And also thank you all our other members from Drone to Phenome, Dave Lebauer, Jennifer, and Max Feldman. Thank you for also helping us this season. Yeah, and if you are over there and you want to be part of this group, as Claire was saying, contact us. Next year, we are looking for new members to take over and also starting organizing these workshops. So send a resume and some information about how that is important for you. And we're going to read these and take care. Soon we're going to be in our social media posting more information. And
also, we probably shouldn't miss out on thanking the viewers who come up every week and watch the videos and watch
Definitely.
The platform wouldn't be anything without the viewers. So thank you for everyone who asks questions and gives feedback. It makes it feel much better for the speakers to have questions and feel like they're not just talking to the camera. So you guys make it a great experience.
Perfect. Help us sharing this video and hope to see you next year in the next season.
All right. Bye.
Bye-bye.